this evening. The format for our seminar this evening is a 45-minute talk, followed by 15 minutes of questions and answers. For our friends online, please use the Q&A button at the bottom of your screen to be able to ask questions. We will come and read those. For those of you in Kiggins this evening, raise your hand. I will come around with the microphone. If you ask your question without the microphone, none of our online friends would be able to hear it. So we try to do that so that everyone can hear. This evening's speaker is Dr. Taylor Lindsay. Taylor is beginning her postdoctoral research on temperate coral energetics at Tufts University this fall. Of course, the document did not fully come up. Taylor first came to Shoals as a high school student. While in college, she served as an intertidal intern here at Shoals in 2017. She graduated with her bachelor's degree in environmental science from the University of New Hampshire, where she conducted work on tree hydrology and the benefits of multicultural and multidisciplinary work in environmental sciences. After spending the summer at Shoals as the lab coordinator in 2019, Taylor moved on to her PhD at the University of Rhode Island. There, she studied the effects of light on tropical and temperate coral biology. She also spends much of her time working to improve equitable access to the sciences and fostering inclusive pedagogy in her classes. She successfully defended her dissertation this spring and completed her PhD in biological and environmental sciences in May. This is a congratulations to Taylor. That's a big round of applause. She already has 3 publications from her graduate work, one in marine ecology, one in limnology and oceanography, and one in reef encounters. I really love the name of that. While at URI, she earned the Graduate Teaching Excellence Award twice. This summer, she co-taught the Shoals Marine Lab's underwater research class, and she's currently serving as a mentor to the intertidal transect studies interns on Appledore, and this is the 3rd year that she has served that role. This evening, Taylor will be presenting Astrangia Things, Unraveling Temperate Coral Symbiosis Using Isotope Ecology. Please join me in welcoming our very own Dr. Taylor Lindsay.
Hi, everyone. Thank you for the great introduction, Sarah. Today, I'm going to talk to you about 2 of my PhD chapters, which are both about temperate coral. So we're not going to talk tropicals too much, but if you want to talk about tropical corals, I would love to chat with you. I just want to say we had a little bit of technical issues, so a few things on my slides are a little wonky, but that's just quick. So we're going to roll with it. As Sarah mentioned, I've had a pretty long time, a lot of experiences here on Shoals. Just as examples, I've been here every year since 2014, except 2016, this one, doing a lot of different things in a lot of different environments on Shoals, including the kitchen environment. So this place means a lot to me, and this is also my first invited seminar, so there's something very special happening. So what are we going to talk about? I have 4 goals tonight. The first is I just want to give a basic intro to some coral bio that's going to help you understand what a normal tropical coral is like, so that when I talk about my temperate corals, you'll understand how weird they are. The coral we're going to talk about is Astrangia populata, which is why we went with Astrangia Things as the title. I'm also going to convince you that isotopes are super cool. Trust me, they're cool. And then we're going to look at a little more deeply at one of my research chapters, does light affect the trophic ecology of my temperate using isotopes? Okay. So when you think of a coral reef, you probably think of something like this, very, very colorful, very beautiful. Coral reefs are really important for people. They have a lot of cultural and spiritual significance. They do medicine, tourism, coastal protection, food. They also provide reef structure, biodiversity, primary productivity, and recycling use. They're really important ecosystems. But there are a lot of threats. And although we're not talking about tropical corals much today, these are all things that will also threaten my temperate coral. So ocean warming, invasive species, seas pollution, marine debris, eutrophication, which is caused by excess nutrients, ocean acidification, sea level rise, harmful fishing practices, anchoring and tourism, and overfishing. There's so many things going on with coral, right? Sunscreen, I didn't even. Okay, so like I said, we're going to talk about the basic coral bio. So when we talk about a coral, there's sort of three main parts we think about. The first is a skeleton, right? That's what washes up on the beach. That's what you pick up and think is pretty, right? That's most likely all you've ever seen unless you have an aquarium or snorkeled in the water, right? Living on top of that skeleton is a coral animal. They are animals. And they have tentacles in a ring like a sea anemone. It's basically a colony of sea anemones that all kind of work together as one. And they feed through that net. And then finally, there are algal symbionts. I'm going to call them symbionts for the rest of this talk. They're algae. They photosynthesize. And they live inside of the coral tissue. And so, as I already mentioned, there's two ways that corals get food. From photosynthesis, from sunlight, and then also from heterotrophy, from feed, right? And in corals, this is a really important balance. The symbionts and the corals share metabolites, nutrients, waste products, and when one doesn't have enough of something, the other one also survives. And any individual coral exists on a spectrum, somewhere in between using 100% of one or 100% of the other. Most tropical corals are going to be doing mostly photosynthesis, mostly water. That will change in my temperate coral. The other thing that's really important here is that light changes as you get deeper in the ocean. We need light to photosynthesize. It's essential to the process. As light originates at the top of the ocean and then goes down the water column, it interacts with water molecules, plankton, anything that's in there. And not only does it get darker, but it actually gets more worse quality of light. Different wavelengths when we go so deep. And as an example, here are two pictures I took in Rhode Island that show how different the environment looks in terms of light. So not only is it much darker at 50 feet, but there's not a lot of color, right? It's really just a couple different wavelengths getting down. And if I took a flash picture in that same spot, it would look weird. It would actually have a lot. And light is really important for corals. If they have too much light, they get stressed out. It's too much for them and they can't handle it. If they get too little light, they won't get enough food. Right? And so somewhere in the middle, there's something just right, like Goldilocks, the amount of light. Okay, so that's all kind of tropical coral, what we expect from a normal tropical coral. But now I'm going to introduce Astrangia paculata. This is the northern star coral. You can see its range is pretty extensive, but it is extremely common in some of the New England states. Its range ends at the elbow of Cape Cod, for now. This is kind of what it looks like. So it grows in all sorts of different places, all sorts of different sizes. Sometimes it is out feeding, like in that top picture, and sometimes those polyps will fit all the way in. Okay. Why do we care about this little temperate coral? Because it does this. So this species uses facultative symbiosis, whereas tropical corals really, really need light, really, really need that symbiotic relationship to get food. The temperate corals are kind of good either way. So they come in these two different ecotypes of color. The white one is what we call aposymbiotic. It doesn't really have symbionts. It's not photosynthesizing. It's getting almost all its food from heterotrophy. The brown ones they have symbionts. We think they're getting food from photosynthesis. And so theoretically, they're more like a tropical coral, food and photosynthesis. Okay. All of my work happens in Rhode Island, so I just want to acknowledge the location where I took all of my samples from. This project was conducted on the lands of the Narragansett and Atlantic people as benefactors of a history marked by racism, genocide, enslavement, and the forced removal of indigenous nations, which were instigated by colonial settlers and are sustained across generations into the modern day. We all have a responsibility to acknowledge these injustices and continue to fight for the visibility, equality, and reparations for modern indigenous. The Narragansett people are incredibly active still in this area, and so one thing I want to do in the future is really work more on that relationship. It's really cool to be able to work in your backyard and interact with the community as you're doing. Cool. So The first thing I wanted to know, because my PhD is on how does coral and light interact? How does Isotope interact with light in this natural environment? So we went out and we did quadrat sampling along a depth gradient from 0 to 25 meters, which is about 80 feet. This work is published if you want to go look at it. And so to explain what I found, I'm going to slowly fill in a depth gradient as we go down. So on your left, there is a rocky reef, if you can imagine, and we're going to slowly go down. And on the right, there's a graph that will open. So in the really bright, shallow areas, this is what it looks like. That top picture is taken kind of close to the surface, and then as you get a little bit lower, maybe around 10 meters, it starts to look like the bottom. In the top picture, you can't see any coral. In the bottom picture, there are two little corals hidden in there. You see the white one pretty well. And then on the right-hand side of the picture, there's a brown spot. That's a ground coral. So you can see they're covered in algae in the shallow. And then the algae starts going away, and you start seeing more of a coral actually exposed to light. Okay. So we call this the macroalgae zone because there's macroalgae zone. If we go a little bit deeper, where there's no more algae, it looks like this. This is most of Rhode Island. It's pretty crazy how much individual tiny little corals there are all over. And you can see in this picture, there are both brown and white living in coexistence. That's pretty much all that's there, at least at my place. There are more white ones than brown ones. And as you go deeper and deeper within this little zone where there's no algae, you get fewer and fewer of the brown. We call this the coral zone. It's a coral. And then finally, if you keep going down, there's just a lot more of this white coral. There's no more brown. And so we call this the deep zone. I don't know how deep they go. This is a question we still have. I don't have the ability to scuba dive that deep. So that is one question I'm posing. And so if we look at the interactions between macroalgae and the coral, as well as light in the coral, we see some really, really strong relations. I'm not going to dig into these graphs too much. What we really took away from this is this picture. Why is this happening? If the symbionts need light to survive, how come if there's enough light to photosynthesize in this area, how come every single coral is brown? Like from an evolutionary standpoint, from a physiological standpoint, we shouldn't have these mixing of two different kinds. And the first question anyone will ask, if they know what to ask, is are these two species? Are they genetically distinct from each other? And the answer is no. At least at this site, we haven't found a difference in terms of their genetics. Okay, so to try and answer this question, I asked, does light availability actually change the trophic ecology of these? So in order, if I change the amount of light they get, is that controlling whether they're brown or not? That's the basic question. This paper was actually published last week or 2 weeks ago, something like that. So it's very new. Thank you. This is what we're gonna think about for most of the rest of the time. So we set up a shading experiment in the ocean. We took about 100 browns and 100 whites. We brought them to the surface in those buckets like you see, and we deployed them back in the ocean in what I'm calling cages, although they're not actually a cage. And we had shades on. So in the shallow site where they came from, there are controls. And then we also put them in the site just to see if there's something going on with depth that isn't necessarily just about light. We of course tracked the environmental conditions throughout the entire time, and we found that the control was always brighter and hotter than the shade, and the same is true of the shade position. Okay, so there's a really nice gradient of environmental conditions. We looked at whether they survive. Always question one when you're doing coral transplant. If you transplant tropical corals, they often have like % survival rate. We had only 4.3 of them. I think that was like 7 corals. So first point, they survive really well, even when you put them somewhere crazy, really deep, really bad. The other thing we noticed is that the 3 different lights, so the 3 different purple colors, had no significant difference. So the amount of light they had did not matter in survival. But the browns and the whites did have a disk. So the brown ones, the ones that have the ability to do photosynthesis, had a significantly higher survival. So something's happening where the symbionts maybe are contributing to that survival rate. We also wanted to see if their physiology changed in the different light environments. Okay, so we quantified the density of their symbionts and also their chlorophyll A. So chlorophyll is what makes plants green. It's a pigment that allows plants and algae to do photosynthesis. Okay, and these are like the most beautiful graphs you could ever imagine. They do exactly what you want them to. Both of them go down with depth. So the with this amount of light. So the darker it gets, the less symbiont and the less chlorophyll. Okay, this is such a clean pattern. So light is changing their physiology for sure, no doubt about it. We also tracked if any of them switch types. So you can see the three different treatments that we had: our control in bright light, shade in medium light, and the dark one. In the control, there was some switching, but for the most part, we started off with half brown, half white, and we ended with half brown, half white. In the shade, where it's a little bit darker, had a few, about 23% stay brown the whole time, keep their symbiont, but a lot of them became aposymbiotic. In the deep, really, really dark area, they all turned white. And this makes complete sense to us because this is deeper than we would see brown ones in nature. So it aligns with that first project that we did. So now I'm a little confused because I now know that light matters. It changes their physiology. It makes them switch to type. But in the wild, we still have this phenomenon where we're seeing the brown ones and the white ones together. So something doesn't make sense there, right? Something else is happening. And so we are going to jump into isotope. So very briefly, I don't know how many of you have, raise your hand if you've taken chemistry. So you know all about isotopes. So isotopes are variants of atoms that contain different numbers of neutrons. All you really need to know is it changes the weight of these atoms. Right? If I asked you to bring half of the rock in this pile down to the floating dock, and I didn't tell you which one, you're going to pick the small one, right? If I say, just bring 5 of them, you're going to bring the smallest one because you don't want to carry a whole bunch of big boulders, right? This is the same phenomenon that's happening all around the side of us all the time. If there's a process that has an option for a heavy atom or a light atom, it is going to preferentially take up the light atom. It'll still get some of the big ones too, but most processes in nature, from evaporation and natural environmental processes, but a lot of biological processes, you have this preference for light. We call this isotope fractionation, and that's as fancy as I'm going to get with it. In our study, we looked at the ratio of nitrogen and the ratio of carbon, and we asked how many times, with nitrogen, we can ask how many times has this food been eaten? So from plant to primary consumer up the food chain, how many times has it been? With carbon, we can ask where did the food come from? Is it terrestrial? Is it marine? We can get even more specific. Okay. Now we get into the isotopes. So I love isotopes. I think this is pretty obvious. And I'm going to give you 20 reasons why you should too. And I'm going to do it in 2 minutes. I'm going to freeze. Okay? So I'm going to go really fast. Are we ready? We have to start over. If you want to learn any more about any of the things I'm about to say, we can talk. Okay. Ready, set, go. So most people know about carbon dating with carbon-14 and radioactive. We can tell how old something is. We also can use events like an atom bomb to date the age of a spiriton. We can also use isotopes to quantify carbon emissions. You can see the seasonal and overall trend that's happening there. We can use isotopes to trace the flow of deep, deep oceanic water that's really, really hard to get to, and maps like that. We can rebuild climate records from a single organism's lifetime or from many organisms' lifetime, like the foraminifera that you see, going way, way back into the past. We can track the changes in diet over a single organism's lifetime using teeth or baleen or otoliths or hair. We can reconstruct really complex diets of organisms like this seal that use a bunch of different types of food. We can track large-scale organismal migration using the barnacles that grow on the back of a turtle or a whale or even a buoy. We can track starvation. As the animal consumes its own reserves, we should see a shift in the isotope baseline. We can look at differences in feeding between species that all go in the same area, and we can't figure out where they're going. We can look at habitat shift as an organism changes its maturity and starts eating larger and larger prey. We can look at maternal investment in eggs, in this case, from the early season to the late season. We can look at that. We can track maternal dependence of a baby from its mother as it moves from lactation into feeding on other stuff. We can trace the fate of a labeled isotope that we put in the water and see where it goes. We can look at turnover rates of different parts of your body. We can identify teeth that from veterans to identify where they came from and where they belong. We can trace cotton supply as it moves across the world to see how sustainable it is. And we can look at not quite. We can look at fast food intake based on how much corn or wheat you eat. And we can also verify that people are selling the correct type of fish and the fish that they say. And there you go, there's your check. So the point of all that is there are so many things. Most of those are only stable isotopes and not even labeling or radioactive. I touched on, but there's even more. So they're really cool. And now you look. This is a little bit more complex. There's two main things that we do with stable isotopes. I'm not going to get into it too much. But if you want the cheap, fast way, you do bulk stable isotope analysis, where you just put the whole tissue into a mass spec. You get 2 numbers out per thing, and you can ask a few questions with it. It is very useful for many organisms. Not great for corals. Corals are too complicated for that problem. And so we, in our study, use compound-specific stable isotope analysis. A lot more complicated. The basis here is that we take a protein, we split it up into every single amino acid that's in there, and then we run it through the mass spec. And now instead of getting 2 numbers, I get 2 numbers for every amino acid. And those amino acids move through your body in different ways. One may not do the same thing as another, and so the differences between them allow us to ask really specific questions. The only one I'm going to talk about today is trophic position. So trophic position, going back to basics, starts at a level 1 for a primary producer. Primary producers are algae and plants for the most part. Level 2 would be anything that eats a level 1. So things like sea urchins, bunnies, and insects. So level 2. And then a level 3 would be anything that eats those things. In nature, it's not this clean and pretty. Lots of things are omnivores, eating lots of different levels. But we need to understand approximately where we would put our coral. So if we were to hypothesize about where my coral goes, I would guess our algae is a primary producer. It is photosynthesizing. We know it's getting a little bit of energy back from the coral, so maybe it's a little bit higher than a 1. And then we would think our coral is 1 step higher, right? Because it's getting energy better. So if we were hypothesizing, guessing, this is where we would put our 2. However, when we do the calculation for trophic position, that is not what we get in our temperate corals and populae. We found a trophic position of 2.5 in our coral coast and 2.4 in our symbiosis. Our primary producer, they should not have a trophic position of 2.4. That's pretty crazy. So what does that mean? That means they're getting almost all of their energy from the coral. Basically, photosynthesis is not contributing to this system almost at all. Okay? So to correct our hypothesis, it looks something more like they are really, really close together. If we look into our 2 different questions here, We see that our treatment, our 3 different light, also doesn't affect surface position in the coral host. And we also see that symbiont density also doesn't really change anything. If we had a larger sample size, maybe something would be going on and maybe more of the brown ones, but this is not a super strong path. Okay. So again, we're back to the original question. We thought this experiment would answer the question, why does this happen? Why are we seeing this phenomenon? And it didn't really answer it because photosynthesis is not contributing to these 2 different types of corals. Then the next question is, why on earth does Astrangia have these symbionts in the place? If photosynthesis is not the reason that the symbionts are in the coral, something else has to be doing. So we go through the literature, right? There's a bunch of different other things that may be happening. We, in our present study, we saw that the brown ones had higher survival. Symbionts may be contributing to their survival. We also saw, other people saw that the brown individuals had improved wound healing. They also had more calcification, so they were contributing more into their skeleton and building their skeleton faster. The brown ones often have a higher growth rate as well. They usually have a lower susceptibility to sedimentation and also improved immunity, so they should have more resistance to disease. And then the brown and the white individuals have a bunch of other differences in nutrient cycling, microbiome, gene expression, metabolomics. I can't say which one's better than the other, but there are differences. So basically, the answer to this question that we're trying to get at was not energetics. It's something else or some combination of other things. So that was talked about things we learned today. Isotopes are cool. We also learned that temperate corals have really unique characteristics, and that is part of the reason why I use this Astrangia thing kind of logo. There's a brown and a white. If you know about Stranger Things, right? The upside down, right? We also learned that light has a really strong influence on the ecology and the physiology of these species. As they go down a depth gradient, they change. And their physiology changes through strikingly. But light has no impact on the trophic ecology of species. We still don't fully understand why the symbionts are there, what they're doing. And with that, I want to thank all of the undergrad collaborators who helped me on this project. Every single symbiont count takes at least half an hour, sometimes an hour. So that was a lot of work that a lot of different people did for me, and I'm really, really grateful. We also had a lot of different funders. Some of these are for my other chapters, but I'm really grateful that I was able to do this work. I've had a lot of collaborators, and all those names at the bottom were all my dive buddies. And those 2 projects took a lot of diving. With that, I will take questions.
You had 14 online people, which is the highest number that we have so far. So just wave to everybody and say thanks for coming. Thanks, Mike, Phil, everybody. We appreciate you being here. Questions in the room, those of you online, please use the Q&A button at the bottom of your screen.
So you mentioned in the isotope methods that in the method you receive 2 numbers for every amino acid from one protein. How does that work? Like, and how is the protein split? And can you give me an example of a protein you and your partners used in the study that you worked on?
Yeah, good question. So I may have misspoken. We're looking at all the proteins all together. So I'm not targeting any individual protein. All proteins are made up of some combination of different enzymes, right? So really tiny. I don't know how well you can do that. This is the process for doing a lot of chemistry to break down from a tissue sample to a something that can go into the mass spec. It involves acid hydrolysis, which breaks down the protein. Some cleanup steps, we use chloroform and methanol, really gross, to clean the sample, because if we put cell walls and stuff into the mass spec, it'll just clog up and won't work. And then we do amino acid derivatization that really, really breaks down the amino acids and also gives them end members, for chem, and you remember what that means, that allow them to be fed by the next. And then we put them in the machine, and every single one takes like 45 minutes, and then you have to like heat it liquid nitrogen and stay up till like 10 p.m. It's a really fun process. And every single individual sample I run is $142. So this is a very time-consuming and expensive process. So that's why we don't always do it. If we can answer the question with full statements. So those are like 6.
Do we know anything about what the separate corals are eating out of the water column? Like, could their distribution be some sort of like resource partitioning? Do you know anything about that?
Yeah. So most corals basically don't have like they're just taking whatever they can get. They do consume like particulate organic matter. Basically if it falls into their mouth. Some predators are better at hunting than others, and we don't have a super great answer to your question. But whether you live in a bunch of algae where like isopods or other fun critters are swimming around, or at 80 meters where all that's around is coral, probably makes it interesting.
Sounds like a great master's program.
Yeah.
Thank you for your talk. I was wondering, since we have a little extra time, if you could talk about the tropical corals that you studied.
Oh boy, yes, I can. Okay, so I did 2 projects in Puerto Rico. One was just looking at how corals change across a light gradient. So no experiment at all, just go down, collect them, see what they're doing. And I did morphology, physiology, and isotopes again, the cheap time. And we actually didn't really find depth gradient or light gradient patterns super strong. What we found is that their evolutionary history is sort of 2 different groups of corals. And the 2 different groups really strongly affect their morphology, and that seems to affect their topology as well. So a coral with a big polyp, Monstrea cavernosa, if you know any of them, is going to be more heterotrophic than something with a teeny tiny polyp, like Temperate, which logically makes a lot of sense, but there's actually no evidence for this yet in the open. So that's one of the 2. The other one is a little more complicated. We did a reciprocal transplant, shallow to deep and shallow. And we were looking at cryptic lineages. So we think that the species in deep is actually different from the species in shallow. So we wanted to see if you transplant them, can they survive? And the answer is kind of no, which supports like this hypothesis that they're 2 different things. That's the spread notes.
Hey Taylor, great talk. So 2-part question. One for some of the high schoolers. Can you explain how corals get their symbionts? And also I'm curious because I know down south, the temperate or the tropical coral rather, they look at the different types of symbionts as a sort of temperature resistance proxy. Have you, or do you have any interest in looking into different types of symbionts, temperate versus tropical,
Yes.
and any sort of like advantages, disadvantages of that?
So first question, there are 2 ways that corals get their symbiont. One is from their parent. So the parent makes an egg that has symbionts in it, and then you carry on, which if the parent has really harvested really great symbionts that are really good at like bleaching resistance and stuff, is really beneficial to their offspring. The other way is the egg has no symbionts in it and it gets them from the environment. These symbionts can be just in the water column as cytoplasm, photosynthetic cytoplasm. Astrangia does the second. It has a completely aposymbiotic white egg, and the symbionts come later, which contributes to this like mystery of why some are doing it and some aren't, because if maybe the parent is passing it on, then whether it's brown or white is determined by the parent. But that's not the case. The second part of the question was about the different clades of symbionts. There are different types. Problem solved, there is one kind in Astrangia. There's only one. So we can't do any, yeah, that's not contributing to this at all.
Thank you for your talk. I was wondering a bit about your methods for controlling for or incorporating like stress and changes in pressure in sort of your transplant or your like in lab experiments.
So do you mean changes in pressure relating to depth?
Yeah, or just like other factors that the coral will feel. You know?
Most of what we did in terms of transplanting the coral is very well established protocol. You kind of have to break it off the rock. It will like kill a few of the polyps around the outside. So yes, that stresses them out. This experiment was 4 months long, so I'm not worried about that stress in the initial period lasting a long time. Also, we did not see a really extreme mortality event, which also suggests that there wasn't a ton of it. These corals are extremely hardy and actually really hard to kill. So if you do things that you're trying to get some like mortality, you have to like ramp up the temperature, like dump parasites on them or whatever. Like it's really hard to kill. There was another part of it. Oh, relating to pressure, most organisms only care about pressure if they're airy. So a coral shouldn't really care about like pressure. That's more like fish.
So a lot of times in science, we are expected to have some breakthrough when studies. Can you speak to the significance and importance of having not necessarily an inconclusive study, but to like the elimination of options for explaining a hypothesis?
Yeah, so actually, I guess I didn't touch on this so much, but what I found sort of breaks a lot of what we thought about temperate corals, because for a long time, everyone was saying, we can use temperate corals as a proxy for tropical. That's not true if their energetics are completely different. Depending on the question you're asking, there are some that are still a good model for other things. But I actually presented at a conference and I was so nervous that everyone was going to yell at me because I'm debunking the thing you have assumed is true this whole time, which is that they're photosynthesizing and that's why they're different. So that was scary. That doesn't really answer your question, but that was a really big thing in this project. I think that when you're at this point, you're just excited to look at the next question. So we're going to be doing dynamic energy budgets on this species, which is the next big question of, okay, we know there is photosynthesis happening. We can measure it in the chamber, but it's not going into the coral. So where is that energy going? What the heck is happening inside? So my answer is yes, it's challenging, but it's multifaceted and it's even more at you.
So you actually kind of answered my question with that last one, but I brought up a new one. How exactly would you go about testing the energy dynamic buster thing?
Dynamic energy budgets?
Yeah.
Yeah. So dynamic energy budgets, I'm only starting. I have a 3-year postdoc doing that. So I don't have the answer, but it's a really well fleshed out theory. There are many textbooks and like thousands and thousands of publications that use this method in all different organisms. The basis is thermodynamics and physics theory. So it's quite complicated. We use MATLAB models to like figure it out. And the most frustrating part is that the simpler the organism, the more challenging it is. Because humans, we follow thermodynamics pretty well. We have rules about what temperature we need to be and how energy moves through our body. Something really simple like coral, none of those rules apply. So it's actually really, really hard, and no one has ever used like the specific method we're going to do on a coral. The only cnidarians that it's been done on are them. Okay? Especially because I also have a really complicated coral, not an easy one. So, 3 years though, come back to me.
We now have an online question, starting with Great Talk Taylor. This is Brad Weiler, just to let you know. If you have an aposymbiotic Astrangia in a tank, but provided symbionts in a tank, do they uptake them? And second part, do you think if you seeded a tank with different symbiont clade, that the aposymbiont or symbiont Astrangia would uptake it even though it's not familiar with it?
So I haven't done either. I believe people have watched. So even in my switching, we did have some apos, the white ones turn brown. So yes, I think the first question, absolutely. I think if you seeded them, and you gave them light, right, they would take them. If you didn't give them light, they'd probably eat them. The second question, I have no idea. Most likely not, would be my guess. If the Greviole one is the one that they have, I think is the only one that I think they're probably because it's Breviolum all the way through their range. Even in the tropics where we know there are others around.
Yeah, and lots more questions.
Complicated.
Questions in the room?
You mentioned sunscreen having an effect. Why and how can like one prevent that, or is there a way?
Yeah, so I'm not an expert on that, but sunscreens have certain chemicals. So there's mineral sunscreens and there's chemical sunscreens. Chemical sunscreens have chemicals in them that are toxic. Mineral sunscreens like zinc oxide are much safer. So use a mineral sunscreen, especially if you're in water with coral. Up here, it's probably not as big of a deal, but if you're in like Florida or something, or somewhere tropical on a reef, mineral sunscreen, it's better for you anyway.
Hey, I know you at the beginning kind of touched on like various things that are going on with coral, and I feel like we really hear them that associated with more southern corals. I was wondering if in your research you've been seeing, because you've been diving for a while up here, and if you've been seeing any of those effects that we really associate with southern and tropical corals up here in these areas, even like at greater depths and stuff.
Nope.
Cool.
They're fine. Astrangia is doing all right. And people always ask me that question. That's just one of the reasons we want to understand this species, because it is in the tropics. Problem is, it's not a reef building. So the ones that can make structure are the reef building corals, and those are the ones that we need in order for the whole reef ecosystem to survive. These little guys are just floating rock. And we actually don't have a very strong rationale for it. It's like ecological role in the greater. There's a science there. Yeah. We're working on it. We're trying to figure out if anything eats them. Right now we don't know.
You mentioned that they are found up to, what was it, Cape Cod?
Yeah.
Right now, what's the information? Is there anything that's
So there's 3 iNaturalist observations in Portsmouth, right over there, and it's just a skeleton. So I don't know if that's real. We are expecting Astrangia to come to the Gulf of Maine sometime soon. We have no idea how that is impacting. Again, they're not like, they don't go fast. They probably will be outheated in this environment. But we actually have dive shops that know and are looking for it, at least that tell people. And yeah, so coming soon, the Gulf of Maine, maybe as the water warms, right? It's right now it's a thermal threshold that the Gulf Stream has to be involved, right? The Gulf Stream goes up to the elbow of Cape Cod, and as the Gulf of Maine continues to warm, probably this environment will change.
How does it move?
It doesn't.
So
Yeah.
then how would it come to the
Reproduction. So it throws its gametes up into the water column, and those gametes will float. The larvae of this species can stay alive in the water column for a while. I think it's like 13 days. So if it's here, it can get here, and then it can get there, right? It will move. But not an individual. They are very much stuck on.
it's like a tree?
Anecdote. It has been found in Ireland on a buoy. They grew on a buoy, and the buoy floated to Ireland.
Any other questions? For those of you online, just letting you know that it is like one of the steamiest nights that we've had on the island. So I'm really impressed with the number of, I'm really impressed with the number of questions.
While we're talking about gametes, are these coral that like spawn with the moon or with the tides, or do we know?
They're weird. They're really weird. They kind of just do a dribble all summer. So this species has males and females, and they separately will release eggs and sperm into the water column. We think there's some kind of chemical too. When one goes, the others will go, but it is not like in tropical coral, they all go at once. It is like all summer long, they have gametes, and we don't have a great explanation. It's not temperature, well, it's temperature a little, but it's not like, yeah. So if we want to collect gametes, we put them in a bucket and we make them really hot. We make them like 30 degrees Celsius and they will fly.
Which is not the temperature of the water up here. Any other questions?
I could be totally wrong about this, but tropical corals like all like reproduce at the same time, right?
Yeah.
Ish, kind
Different
of.
species.
Yeah. Is that the same with temperate corals?
At least not this species. It's all summer. So they're going like we collected them in June and we got them to spawn. Maybe they would have waited another month in the wild, but we don't like you can kind of tell when they're about to do it if they puff up. It looks really blobby. Like a sea anemone that's yeah. So you can tell, and then sometimes it just pokes them. But I've only seen that in the wild one time.
Any other questions? Carrie, do we have anything online? No? Well, I want to thank everyone for coming for this evening's Rock Talk. I want to thank Taylor for trying to convince everyone that isotopes are cool and doing so in 2 minutes and 10 seconds, and for giving us such an interesting set of new questions to come about with what's going on with the species that many of us have never heard of before or thought about corals at Cape Cod or anything along those lines. So if everyone would please help me in thanking Taylor for a phenomenal talk. For those of you online and those of you in the room, we have 2 more Rock Talks left in this series. Next week, we have Trixie Betts coming to talk about an aquaculture internship program in the state of Maine. And the following week, we have one of our faculty members who teaches marine immersion, Jason Goldstein, who will be doing the last Rock Talk of this season. Both of those are at 8 p.m. If you're interested, you can join us online. Our Rock Talks are all live streamed. And we also will be live streaming again our entire session next year. So thank you all, and thanks again, Taylor.